Everyone, so I'm back with the second half of the video lecture on digestion. We're just going to pick right up in the stomach. If you're seeing this first, there is a first half that goes from the mouth to the stomach with a little introduction, but let's jump in where we left off. So we had a gathering of foodstuffs that have been chewed and mixed with saliva. They've made their way from the mouth into the esophagus, from the esophagus through the pyloric sphincter and into the stomach. And the stomach is probably the most radical environment within our entire body because of the presence of gastric juices, which is a combination of hydrochloric acid and some digestive enzymes. The pH within the stomach, when it's not actively digesting, is 4 or 5, which would be considered a weak acid. But when actively digesting, the pH will lower down to 1 or 2, which will be considered a strong acid. So the pH of the stomach is extremely low compared to any other region within the body. I mentioned in the last video, or at least I left off with the question of why doesn't the digestive system digest itself? Well, throughout the digestive system, there are layers of mucus that line the digestive tract, and that protection is what prevents the digestion of the digestive tract itself. And those protections are particularly strong in the stomach. So there's a couple things happening. There is the chemical breakdown, in particular of proteins, and the mechanical breakdown of the food through churning. So the stomach is lined with muscle, and that muscle is capable of it's a really odd thing to see. The video that I linked in the previous in the slideshow itself shows this muscular contractions. It's kind of a rolling and grinding motion. And within the stomach, the food is turned over and mixed with the gastric juice, including those enzymes and acids. Proteins are broken down chemically, but that movement of the stomach itself, the churning of the muscle itself, provides a little mechanical digestion as well. The proteins that are broken down by an enzyme called pepsin. And pepsin is the enzyme that breaks down peptides. It's not called peptase in this particular case, which we might expect, but this diagram here shows that dual role that the stomach plays. Food spends probably 4 to 6 hours in the stomach, depending on what's present and how much, which is quite a bit of time. It doesn't take long for some foods to move through the stomach into the small intestine, but there is a time during which the chemical digestion takes place. And here I've just included a slide that is a quick review of enzymes and how they function. Remember, enzymes are about shape. The function of an enzyme is about shape. Sorry if you hear a little rustling here. I'm in the kitchen, and strangely, I have a bag with snake food in it. And not everyone likes to hear this, but the snake food is alive, and they're very active. They're making a lot of noise, so I'm just in the kitchen moving the bag to a new spot. Sorry about that. But remember, enzymes are all about the correct shape, and the enzymes that are active in the stomach, the pepsin enzymes, are not active until the pH reaches a particular level of acidity. So when you look at the optimal environment for pepsin, you see that optimal enzyme activity happens around a pH of 2, And then an enzyme that we're going to see shortly in the small intestine, trypsin, which is also involved with breaking down of polypeptide chains. It's also helping to chemically digest the proteins in the small intestine, is most effective at a pH of about 8, so very basic. So you have two enzymes with the same general function in the body. They work in different locations, and they work best at very different pH levels in very different environments. This is a diagram that just shows through the secretion of hydrochloric acid within a gastric gland. So this gland on the left would be pitted in the stomach lining itself. These cells then would secrete hydrochloric acid and the other fluid components of gastric juice. And here you've got the inactive version of the enzyme shown as a polypeptide chain. You have to cleave or cut part of that enzyme away in order to activate it. And what makes that possible is the hydrochloric acid. So the inactive version of the enzyme is called pepsinogen. In the right environment, which is created by the secretion of hydrochloric acid, the portion of the enzyme that prevents it from working is actually cleaved off, cut off, and then the pepsin is activated and breaks down polypeptide chains into smaller segments of amino acid chains, or even individual amino acids. Here's another diagram showing the same process, but this diagram includes then the transition between the stomach and the small intestines, in which further breakdown of proteins takes place with the enzyme trypsin. Trypsin's the enzyme that I would like you to be familiar with. They mention chymotrypsin as well, but trypsin works. Essentially two enzymes with the same function working in very different environments, and that they work in a sense in sequence is very interesting. We cannot completely break down proteins in the stomach, but we're able to begin that process, and then the process is finished in the small intestines using a different enzyme under very different conditions. Here's a look at the digestive tract and the pH levels that are measured along that tract. And if you look from esophagus to stomach, you see a highly acidic environment. Then as food moves into the small intestines, we get more of a neutral environment. And then we actually get into, in some portions, the darker greens are actually bordering on basic. So you can see the pH levels change throughout the digestive tract, and that change, that variation, is probably going to be tied in most cases to enzyme function, because enzymes, as we know, prefer very specific temperatures, very specific pH levels. Just a word about that protection that I mentioned. The lining in the stomach is particularly thick, particularly effective, especially because it has to be dealing with a harsher chemical environment. And the lining within the stomach is even more protective than it is elsewhere. That lining is called the mucosa, and the mucosal lining can break down on occasion, can thin, and then the acidic environment, the gastric juices within the stomach, do in fact begin to break down the tissues beneath. If the lining wears away, and you can see in the photograph, it's not very pleasant to look at, but that is a gastric ulcer, an ulcer of the stomach lining. And an ulcer is essentially a region of tissue that has been exposed to gastric juices and is damaged by them. So you can see what damage would occur without the mucosal lining. If you have felt heartburn, then you are experiencing the backup of some of the gastric juices into the esophagus. And so you can see that's very uncomfortable. And the pain that you feel, the discomfort that you feel, is an indication that those gastric juices are very harsh and causing mild damage. They're backing up through the pyloric sphincter to that lower esophagus, maybe because you've overeaten. And of course, again, not pleasant to think about, but when we vomit, there's a very harsh experience of those gastric juices as they come up through the esophagus into the mouth and then out of the body. So again, not pleasant to think about, but we all have had experiences with how intense the gastric juices are. This slide is really just to dispel a myth, in case you're still hanging on to that myth, that stress creates ulcers. Ulcers are caused, the weakening of the mucosal lining is caused primarily by a bacterium, and it's Helicobacter pylori, or H. pylori. And there you can see the official scientific name, capitalized genus, lowercase species. That's the scientific name. The lining of the stomach can also be weakened by anti-inflammatory drugs, specifically the non-steroidal variety. And the most commonly used version of that is probably ibuprofen. So I know when I had my kidney stones and needed pain relief regularly over a period of many weeks and didn't want to take the more effective, the more intense pain relievers that were provided because of their side effects, I took a lot of ibuprofen, and probably more than I should have, and I had a really, really upset stomach. So that stomach upset is related to the interference that these anti-inflammatories create in the stomach lining itself. So not a good thing. I think you're supposed to, and I'm no expert when it comes to medicinals or pharmaceuticals, but I think you're supposed to alternate between Tylenol and ibuprofen in order to minimize that effect. So at any rate, there is a bit about how the stomach protects itself and the specific environment within the stomach that is needed in order to begin to digest proteins in particular. Now, the material in the stomach, it arrived as a bolus and is now mixed with gastric juices, has been chemically and mechanically or physically modified. And as it passes from the stomach through a sphincter into the small intestines, we refer to it as chyme. We refer to it as chyme from this point forward. Probably would not be a very pleasant sort of stuff because of the acidity initially, and by now the food is very soupy and has begun to break down, but it's nowhere near complete. So the chyme, the digesting food, is passed from the stomach through a sphincter into the upper or initial section of the small intestines, which is called the duodenum. And we're going to see that the small intestines is separated somewhat artificially into three sections. I mean, there is no physical marker between these sections. We've sort of imposed them, but there is a sort of a change or a sequence of events that take place in the process of digestion. So we've divided the small intestine into these three sections, sort of based on what's happening to the food or what's primarily happening to the digesting food along the way. So the duodenum is the first section, jejunum is the next section, and the ileum is the third section that we'll see. So I'm calling this the small intestine, a tube in three parts. The sphincter through which the food passes. Remember, it was there was a sphincter at the entrance to the stomach, and there's a sphincter again that is sort of managing the passage of food between the stomach and the small intestine, the pyloric sphincter. There are some important contributions now from some of the accessory organs, namely the pancreas, which has a number of functions in the body, but in terms of digestion, secretes digestive enzymes that and other fluids that are involved with further chemical breakdown of the food and also with the neutralization of that acidic chyme. So the chyme, remember, coming out of the stomach is going to have a really low pH, but it needs to be neutralized in order for the enzymes that are going to help with this continued digestion to do what they need to do. There are pancreatic enzymes and other fluids that help to neutralize the chyme. In the liver, so that's the pancreas. In the liver, you have the production of bile. Now, the liver has a number of functions in the body as well, but digestively, it produces bile. And bile is produced in the liver and then stored in the gallbladder. And you can see the arrangement of these organs here in this diagram. Bile is very interesting. Bile is not involved with any chemical digestion of the food. Instead, it's further mechanical digestion. And bile is a little bit like a detergent. In fact, it may even be officially classified as a detergent. So if you think about what a detergent is good for, it's good for dealing with oily, greasy messes, right? And if you put a little oil and water together, the oil is always going to gather at the top. But if you put a drop of, say, dishwashing soap in with the oil and water and seal a container and shake them up, the oil will break into tiny droplets. And those droplets won't regather into a layer of solid oil at the top anymore. They'll remain separated from each other because of the presence of the soap molecules, the detergent molecules. And we don't have to get all the way back into the hydrophilic, hydrophobic idea, but we saw this happen when a drop of soap broke the hydrogen bonding at the surface of water when we did our activity with surface tension. And a detergent will, when oil or grease are present, will break the material into smaller parts. Now, what that does is simply give enzymes more surface area to work on. You know, the bile will help to break fats and oils into smaller droplets within this liquid chyme, and the enzymes at work on those fats in order to break them down into their component parts, namely fatty acids and glycerol, are going to have access to more molecules if they are organized in, say, millions of tiny droplets instead of one layer of oil. So it's a mechanical aid, it's a mechanical benefit that bile produces. Here are gallstones. I thought we would just take a peek here. There's so many different ailments we could focus on in looking at digestion, but I thought we would at least look at a couple. Gallstones, probably everybody knows somebody who's had them. And they're basically hardened bile salts, and they form in the gallbladder. And if one of them comes loose or is dislodged and moves towards the bile duct, which is going to be the duct through which bile is transferred from the gallbladder into the small intestines where it has its effect, then the gall duct can become, or the bile duct rather, can become infected and inflamed. It's very painful. So gallbladder surgery is fairly common. You can remove the gallbladder without any undue harm over long terms to the individual, and you can get the gallstones out. So the photograph is of actual gallstones in a gallbladder that has been removed from a patient. So it doesn't look very comfortable, but that's what gallstones are. They are essentially bile salts. And the exact chemistry of how liquid bile hardens into salts, I'm not entirely familiar with. But I imagine it's a bit like anything else that's in solution. For example, we have hard water in our home. Maybe you do too. So over time, all the faucets get sort of a crusty, salt-like mineral buildup, and I imagine the buildup of bile salts is something like that. So there we are. So we're in the second part now. We're also we're still in the first part. We're still in the duodenum. So we're still in the very first section of the small intestine. We need to mention that there is the breakdown of starches and sugars, along with the breakdown of protein by trypsin, which we've mentioned, and an important new process begins, which we knew was coming, which is absorption, right? So the absorption of fatty acids take place here as well. Okay, so we're still in the first section, the duodenum, which is in blue here in the diagram, just after the pyloric sphincter. The jejunum, and this is where the majority of nutrient absorption takes place. So I'm not willing to say with my limited knowledge that there's no further breakdown of food, that all the chemical breakdown of the food has taken place, and all that's left to do is to absorb what we want and to leave behind what we don't. But because there is so much absorption that takes place in this next segment of small intestine, we want to look at a structure and function idea that allows this to happen more effectively. So if you look up close at the lining of the jejunum, and perhaps all along the small intestines where absorption is going to take place, you will see that there are these tiny little extensions. And what these tiny extensions essentially do is increase the surface area. And that makes sense because when you're absorbing nutrients, you want to absorb as quickly and as effectively as possible. These little extensions are called villi, and a single extension is called a villus. So the villi are then covered, of course, with epithelial cells that are well suited for absorption of molecules that have been freed from our food. And then the villi are covered with microvilli. So the cells lining the surface of the villi have tiny extensions, as they show here, that even further extend the surface area for absorption. So think of the jejunum and the small intestine in general as being about further chemical and mechanical breakdown of the food into its molecular components, but then the really crucial role of the small intestine that definitely differentiates it from the stomach in terms of functioning is the absorption of these nutrients. And the structure, the microstructure that allows for the most effective absorption is the inclusion of these villi and microvilli to increase the surface area along the inside of the digestive tract. So at this point, we move on to the third section of the small intestine, and there are a number of other important components that need to be absorbed. We need to absorb vitamins. Everybody's heard of vitamins and minerals, and we need to get those molecules into the bloodstream where they can be circulated to the cells of the body, just like all the other important nutrient molecules. Vitamins are essentially molecules that serve a particular function, usually biochemically. Many of the vitamins that you're familiar with are important for enzyme functioning. So enzymes are often activated by a molecule, and that molecule is at times classified as a vitamin. And there are a number of them that are crucial. The actual biochemistry is complex, and most of it is beyond my understanding. But these vitamins become important. And what's really sort of fascinating is that many of these vitamins can only be created by the bacteria and archaea that live in our gut. So they're derived specifically from bacteria and archaea that thrive in the gut, and that is one of their contributions. They are capable of freeing the molecules in the process of obtaining their own nutrients. It's a byproduct of their biochemistry, and we benefit from the presence of those vitamins and other molecules. So if you remove all the bacteria from our gut, we will be vitamin deficient. That's the bottom line. Bile gets reabsorbed in the ileum as the small intestine sort of comes to a close, and some water is also absorbed at that point as well. Here's another look at the villi, and what I wanted to emphasize with this diagram, it's a really nice one, is the close association between the circulatory system and the villi. So absorption is taking place, and of course we want to get absorbed nutrients into the blood so that those nutrients can be carried and diffuse into every single cell in the body. So this is where nutrients from digested food are eventually going to end up in our fingertips and in the cells lining the back of our eyeballs and in the cells of our brain and so on. Those locations in the body are not directly connected to the digestive system, but they are indirectly connected to the digestive system through the circulatory system. The green set of tubes is the lymph system. We'll talk more about the lymph system when we look at human immunity. So it's just another series of tubes that carry fluid throughout the body, but not nutrients and not blood. So you can see just this folding upon folding upon folding. We're getting lots and lots of surface area out of these villi. I think it might have been in a previous slide. It might be in an upcoming slide. I don't remember. But the estimate for the actual surface area within our small intestine is something close to that of a tennis court. So if you could smooth out all of these folds and all of these villi and all of the microvilli into a flat sheet to actually show how much surface there is within the small intestine for absorption, you would end up with a sheet that's about the size of a tennis court, which is pretty amazing. Keep in mind that the small intestine is some 25 feet long. So it's a long tube with all these folds and villi and microvilli, and all of that provides a lot of surface area for absorption, which is, of course, a really key step in this whole process. This is a slide devoted to our microbiome. We could do an entire week on the microbiome just because the organisms that make up the microbiome are prokaryotes, and we would get to see the diversity in prokaryotes. Remember, there's simpler cells that lack organized nuclei or other organelles, but the bacteria, as the most well-known example of a prokaryote, are extremely diverse, and in some cases there's as much genetic diversity between bacteria, different groups of bacteria, as there is between all of the animals or all of eukaryotes for that matter. There is a lot of genetic diversity and evolutionary history in the phylogenies that we're able to discern for the bacteria and other prokaryotes. So very amazing organisms, absolutely crucial to our health and survival. We are, in a sense, made up of our own cells, but also a number of other cells. And we get from them so many important digestive molecules, molecules associated with nutrients that we would not get in any other way. So entire books have been written about the microbiome. I feel like I'm really not doing it justice here, but keep in mind that the microbiome, the actual bacteria that become part of our digestive tract, that end up living on our skin, in our mouths, in the lining, and occupying the fluids within our eyes, they're everywhere. They're picked up from our environment. So in utero, they're picked up from the mother. Even after birth, well, especially after birth, because of the close proximity between the mother and the newborn babe, there is a lot of transfer of bacteria and other prokaryotes, the archaea, that make up our microbiome. We can get bacteria that are unhealthy as well. And in fact, some of the bacteria that do us the most benefit become unhealthy if they increase in number. So this balance of microorganisms in the microbiome is increasingly understood to be very important. So lots to know, but I just want to at least mention, you really can't talk about digestion without thinking about the microbiome, even if just for a few moments. Absolutely crucial in the process, absolutely crucial for our survival, and we're finding out more ways every single day about how crucial the microbiome is. And why it varies over our lifespans, and how it changes with environment and with diet and a number of other things. So there it is. Okay, large intestine. We got to move our way along here. The large intestine begins down here on the lower left. It would be the lower right in our actual body's lower left in the diagram. So food has left the stomach, made its way through 25 feet or so of small intestine, and then the chyme is passed on into the large intestine right here. So this passage, there's a sphincter there to help control that, is important because we're not done with the digestive process yet. We have essentially absorbed all the nutrients we're going to get out of the food. So the large intestine, as far as I know, is not going to be involved with any absorption of nutrients, but is involved with the absorption of water. So hugely important to regain water back from the chyme. It's been important to kind of keep things moving, to keep things soupy, to keep digestion and absorption taking place as effectively as possible. But we need to get that water back. And the large intestine is divided into three parts: the cecum, the colon, and the rectum. Again, it's not always easy to tell exactly where these particular structures are. They vary in shape a little, but essentially the large intestine can be divided into three sections that are more or less serving the same function for our purposes. Okay, so a majority of the nutrients are gone, if not all of them. I hesitate to say all and always. It's just usually you're going to find exceptions if you do, but for our purposes, the nutrients are gone and we need to get water back. That's what we're primarily doing. So water is absorbed from the undigested material. What's the undigested material? Well, it's going to still be largely water by the time it's passed as feces, but there's also bacteria that are going to accompany undigested material. The undigested food material would be largely what we call fiber, so it would be plant material that can't be digested. And so that's what's going to pass into the small intestine, and that's ultimately what's going to make up the majority of the feces as well. At this point, we essentially have feces, but it's too watery. So we get that water back, and we make an attempt to prepare the food for elimination. Also, at this point, we see the appendix. The appendix may, as we learn more about these microorganisms, may serve an important function in helping to maintain our microbiome, but at this point is largely considered vestigial. You might remember vestigial structures are modified or remnant structures from our evolutionary past. The appendix is a much larger and more obviously functional structure in other vertebrates, but not so in humans. At any rate, it may eventually end up to serve some important function that we just don't know about. In this particular case, the ailment or the particular problem I thought I would mention is, of course, appendicitis. So if you get blockage right here at the duct leading into the appendix, then bacteria, the wrong kind of bacteria, can proliferate. You can get infection, and that infection then becomes extremely painful and, in fact, very dangerous, because as the bacteria grow in the appendix, the appendix can burst, and then you've got bacteria, unhealthy bacteria, that then are released into the abdominal cavity, and that infection can spread and can even cause death. So it's important that if you have pain in your lower right abdomen, that you have it checked out in case it's appendicitis. They can remove an appendix, like the one you see here, very inflamed and swollen, but they can remove an infected appendix and suture the opening that's left behind without any undue harm over longer periods to the patient for whom that's done. Okay, so I think I've mentioned all of this. We're moving towards the rectum and the anus. The anal sphincter, of course, is the opening to the outside world through which feces passes and during the elimination process. And the rectum is the last portion of the large intestine in which feces is stored until it can be eliminated. So there's some mention here of what we find in feces. Of course, the composition of feces is going to vary depending on what we eat. If you have an infection, and generally anywhere along the digestive tract, beginning with the small intestines, but if there's an infection, then basically what the body does is it fails to absorb the water. In a way, it's a way of speeding up the process of elimination to get the infectious bacteria out of the body. So that's what diarrhea is. When we don't absorb the water into the large intestine and the body in an attempt to very quickly eliminate. And think about what happens when you're sick. You get diarrhea, you might vomit, especially if you get something like food poisoning, which is essentially a pathogen that enters the digestive tract with our food. And everything that happens from stomach upset that results in vomiting to very quickly moving material through without a whole lot of digestion or absorption taking place, diarrhea is just a way of ridding our bodies of the infectious agent as quickly as possible. The other disease or ailment I thought I would mention in this context, of course, is colon cancer. So this shows the development of polyps, cancerous polyps. And they can grow into tumors. And the danger there, of course, with any cancer is that cancer cells slough off of the tumor and are then transferred to other parts of the body. So metastasis is the process by which cancer cells move from the original tumor where they grew and developed to another part of the body and begin to spread. And colon cancer is a very common type of cancer in the U.S. It's some statistics around that particular cancer are included on this slide. The key age that we tend to target for examinations, regular examinations of the colon, or at least tests that can detect the possibility of cancers in the colon, is 50. But I've done some reading this week that's basically said we need to back that up, that there's an increasing number or incidence of colon cancer in younger people as well. So there's some statistics there. Statistics about cancer are tricky because, cancer is going to vary of course, depending on diet and depending on weight and depending on genetics and depending on your environment. There's so many factors. But colon cancer is, in fact, more prevalent in the developed world. So there's a lot of evidence to suggest that our modern and Western diet contributes to the incidence of colon cancer because of the highly processed foods that are so common and the high amounts of fats and others. So we don't have any easy answers to how to prevent colon cancer other than to do the things that we're told to do for everything else that might ail us, which is to watch our weight, to exercise regularly, to eat well, to get lots of sleep, and to limit, certain foods and drink that we know can be harmful to the body in a number of different ways. Probably a good idea, not just for the sake of limiting our chances of colon cancer, but for limiting our chances of encountering a whole lot of other health challenges. So colonoscopy. So this is a diagram that shows how a colonoscopy works. And this is where basically a surgical tube and camera are inserted into the colon through the anus, up through the rectum, and then can be used to search for polyps, whether they are cancerous or not. Generally, they can be removed in the same procedure. So the tool is both a camera and a polyp removal tool. And regular colonoscopies are encouraged for everyone over 50. They also, I know that Kaiser, for example, sends out a test kit in order to minimize the number of colonoscopies that are performed or needed. The test kit involves taking a sample of feces and putting it in a solution, sealing it up in a capsule, and literally mailing that sample back to a lab that tests for certain cells, in the presence of certain cells and maybe proteins as well. I don't know the details of the test that's run, but at any rate, if there is a indication that there may be polyps developing from this test, then you go in for the colonoscopy. I've had friends who are practicing doctors say that their recommendation is just to get the colonoscopy, but it's not a fun process. Nobody likes to do it. So if they say, hey, you can just take a sample of feces, send it into a lab, and that is an effective screening tool, then a lot of people will be attracted to that. And I know for a fact that Kaiser uses that screening program, and other practitioners may as well. All right, so this is just an overview slide. I'm not going to spend a lot of time there. You can take a look at that summary slide, and we end with a vision of all kinds of wonderful food. Food is a really important part of our biological lives, but it's also an important part of our social lives, our emotional lives. So, making wise choices with food is something that we've all been encouraged to do. It's difficult. I still enjoy a Pop-Tart from time to time. I still enjoy a glass of whiskey from time to time. I still go to Panda Express, though I know it's loaded with all kinds of processed stuff and sugars that I shouldn't have on a regular basis. So I'm not here to give any nutrition advice, but only to invite us all into a little deeper thinking and maybe some research about how we can make choices that would prolong our lives and keep us healthy. So there's an invitation to myself, and I share that with you too. I don't have easy answers, but I do know that there's a lot of information out there, and we can still enjoy food in all the ways that we do by making healthy choices at the same time. So, all right. So as always, reach out with any questions or concern. And I look forward to seeing you very soon. Take care. Okay. Bye-bye.